Hi everyone, welcome to the second part of case oriented revision biochemistry for NEET. Today I will be discussing about some new trend questions in the carbohydrates and the must learn topics before exam in the carbohydrate topics. We shall discuss the question now. The question number 1 is, a 18-year-old sign artist is worried about the weight gain. So, she is consulting a dietitian and the dietitian advised low calorie diet. So, different options are given here. The question is, among the following option which has the lowest calorie. So, the 4 options are given here. So, in order to answer such question, we should be knowing what is the calorie generated from each and every nutrient. So, I will just give you a diagram for that. So, here we can see that from 1 gram of carbohydrates 4 kilocalories, from 1 gram of protein 4 kilocalories, from fat we will get 9, maximum is from the fat, and from 1 gram of alcohol we will get 7 kilocalories. So, based on this we have to find out what is the total calorie generated. So, 100 gram of protein means that is coming 400 kilocalories. 100 gram of fat means 900 kilocalories. 100 gram of carbohydrate means so likewise I have calculated it here. So, the first option we will get 1700 kilocalories. From the second option of second option we will get again 1700 kilocalories. From the third option, if you calculate, it comes to about 1625 kilocalories, and the fourth option comes to about 1550. The question was lowest calorie. So, which is the option with the lowest calorie? It is D. So, this is how we have to calculate the calorie that is generated. Now, we will see the next question. A physical trainer advised low calorie intake and to do exercise. The trainee decided to fast for 48 hours. So, comment about the activity and the covalent modification of the given enzyme. So, what we should understand is that this is a stage of fasting. So, basically we know that there are 2 stages, well fed and the fasting stage. So, in the well fed stage the hormone is insulin and in the fasting stage it is glucagon. So, the enzymes which is active under the influence of insulin is active in the dephosphorylated state, whereas the enzyme that is active under the influence of glucagon is will be in the phosphorylated state. So, the aim of the well fed state is to decrease the blood glucose, whereas the aim of the fasting stage is the different hormone in the fasting stage is to increase the blood glucose. So, this is the basic concept. So, here we will see what are the options that is given. First is the PFK1. PFK1 is an enzyme in which state it is an enzyme in the well fed state, because it is an enzyme of glycolysis. The second option is pyruvate dehydrogenase, again it is active in the well fed state. The third option is pyruvate kinase, that is also an enzyme in the glycolysis. So, that is also active in the well fed state. So, we know that in whatever is active under the influence of insulin is it is active or be in the dephosphorylated state. So, here all the enzymes that is given here are the enzymes that should be active in the well fed state, that should be active in the well fed state. So, in this question we have to find out the option in which all these enzymes are in a low activity state. So, you can see here the option B is saying that A is saying low, high, then low. It is not like that, it should be all low. So, let's see the option B, it is low, here also low activity, here also you can see it is in the low activity state. And why they are in the low activity state? Because these are the enzymes which has to be active in the well fed state. So, in this stage, like in this particular case, the person is in fasting stage. So, in the stage of fasting, we know that all the enzymes will be in the phosphorylated state. So, the covalent modification here is the phosphorylated. So, P stands for phosphorylated. So, in this state you can see all these enzymes are in the phosphorylated state, hence they are all in the low activity state. So, the answer here is B. Another question is, a marathon runner reaches the last mile of the race but becomes dizzy, lightheaded and confused. These symptoms arise because of which one of the following? So, here we can see that the glucose is not reaching the brain. That is why all these symptoms are happening: dizziness, lightheaded and confused. So, we are going to see what is the answer. So, we will see the options. Enhanced glucose transporter 4 induction. So, glucose transporter 4 induction is happening like in the well fed state under the influence of insulin. So, here nothing like that is given. Reduced blood glucose for glucose transporter 1. That is the correct answer. Why? Because glucose transporter 1 is the transporter of glucose in the brain. So, here that is affected and hence these symptoms are appearing. Now, I will see the next option, reduced blood glucose for glucose transporter 2. But see, you can see here the glucose transporter 2 is not the glucose transporter of the brain. This is present in the liver, this is present in the pancreas, this is present in the intestine. It is not present in the glucose, it is not present in the brain. So, that is not the answer. The inhibition of glucose transporter 5. You know, glucose transporter 5 is the transporter of fructose and that is not a transporter that is present in the brain. So, the question here is, which is the transporter that is present in the brain? So, in the brain the answer is glucose transporter 1 and hence 2 is the answer. The question number 4, the mucopolysaccharide that determines the charge selectiveness in the given diagram. So, we should understand what is this diagram, then only we can answer the question. So, this is the diagram of the membrane, that is the renal glomerular basement membrane. So, we should know what is the glycosaminoglycan that is present in the glomerular basement membrane, which is determining the charge selectiveness. So, we will see the options. The options are heparin sulfate, heparin, keratin sulfate and chondroitin sulfate. So, you know what is it? Yeah, it is heparin sulfate. So, such kind is we should know two things, then only we can answer such question. We should be able to identify the diagram and then we have to find out what is the glycosaminoglycan. And again one more thing I should tell, in the last NEET there was a question based on the diagram. See, rather than giving this heparin sulfate as like that, it was given the diagram of the 4 glycosaminoglycan and we should be able to find out what is the glycosaminoglycan from the diagram. So, we should be thorough with the ring structures of the glycosaminoglycans. Then which one of the following occur during the conversion of pyruvate to glucose? This is also an indirect way of asking pyruvate to glucose. What is that pathway? It is the pyruvate to glucose pathway is the gluconeogenesis. Gluconeogenesis. Now, once we understood what is pyruvate to glucose, that is gluconeogenesis, now we will see the option. All the reaction occur in the cytosol. Is it like that? No, there are reaction that is occurring in the mitochondria as well. Biotin is required as a cofactor. Yeah, pyruvate carboxylase is a reaction and that require biotin. That is also a correct option. Carbon used as carbon dioxide in one reaction appear in the final product. That is not correct because one carbon dioxide is used for a carboxylation reaction, that is pyruvate carboxylase, but that is not coming in the final product, that is glucose. So, that is option is not correct. Energy is used only in the form of GTP. No, energy can be used in the form of GTP as well as in the form of ATP. So, if we see the reactions, I have given a rough skeleton of the gluconeogenesis. The option 1 was saying that is about the pyruvate carboxylase. In this reaction, we know a carbon dioxide is added and biotin is required. So, biotin is required. Then one option was saying a carbon that is added is coming in the final product glucose. No, it is not like that. This 3 carbon compound is converted to a 4 carbon compound by one of the key enzymes of gluconeogenesis. In the next step, oxyl acetate is converted to phosphoenol pyruvate. That enzyme is PepCK. In this reaction, that carbon dioxide that is added here is released. So, it is not coming in the final product glucose. Now, about GTP. GTP is required, but all the reaction is not requiring GTP. So, which is the reaction that require GTP? So, the answer here is biotin is required as a cofactor. Another question, a chronic alcoholic developed a high output cardiac failure. The treating doctor doubted a vitamin deficiency. He advised to check the activity of one of the enzymes in the carbohydrate metabolism. Which enzyme activity is the one that he advised to check? That is chronic alcoholic. They are having the chance to develop thiamine deficiency. That is B1 deficiency. So, if there is B1 deficiency, what is the indicator of B1 status of the body? That is erythrocyte transketolase. Erythrocyte transketolase. So, which enzyme in the carbohydrate metabolism has to be checked? It is the transketolase. Then one more question. That is 7th question. A 3-month-old child is very irritable, fussy and tired in between feeds. That word is very important, in between feeds. He started to develop abdominal distension, hepatomegaly, and blood level of lactate and uric acid is elevated. So, there is lactate level is elevated and uric acid level is also elevated, and this child is having hypoglycemia. So, all these are pointers towards a glycogen storage disorder, and specifically it is the liver glycogen storage disorder because there is hypoglycemia. So, now what is the answer? Glycogen synthase is a liver glycogen storage disorder, but glycogen synthase deficiency usually it is a condition which is very severe, and usually these children die at birth or before birth itself. These children die. That is in utero, death in utero is possible in the case of glycogen synthase deficiency. So, very rarely this can be a cause, very rarely this can be a chance of glycogen synthase because it is a very rare disorder. Then glucose 6 phosphatase, that is the answer here. Glucose 6 phosphatase is causing type 1 glycogen storage disorder, that is von Gierke's disease. So, that is having all these features. Then pyruvate kinase, no, it is not causing this. Then muscle glycogen phosphorylase, no, it is not, because muscle glycogen phosphorylase deficiency will not cause hypoglycemia. So, that is also not the answer. So, the answer here is B, that is von Gierke's disease, glucose 6 phosphatase. The next question is, type 1 diabetes mellitus patient on insulin experienced tremors, tachycardia, confusion, dizziness, and she was brought to the emergency department and the doctor advised IV glucagon and she responded. What is the biochemical reason for this? So, something which is activated by IV glucagon. So, what is it? That is the question here. So, stimulation of glycogen phosphorylase in the liver. Now, IV glucagon, it increases the activity of glycogenolysis. So, glycogenolysis. So, and this glycogen phosphorylase is an enzyme of glycogenolysis. So, that is the reason, and we will see the rest of the option. Release of cyclic GMP. No, it is not the cyclic GMP, it is cyclic AMP that is increased by glucagon. Then stimulation of conversion of glucose 6 phosphate to glucose 1 phosphate, that is also a very confusing option. But we know that in the case of IV glucagon, which is favoring glycogenolysis, actually it is glucose, it is not glucose 6 phosphate, it is given here glucose 6 phosphate to glucose 1-phosphate. But in the case of glycogenolysis, it is the reverse. Glucose 1-phosphate is converted to glucose 6-phosphate. So, now we will do some of the must learn topics under the carbohydrates. In the chemistry of carbohydrates, the must learn will be the glycosaminoglycan and the mucopolysaccharidosis. Then the ring structures of glycosaminoglycan. I have told you the last year there was a question on the ring structure of glycosaminoglycan. So, we should be knowing what is the ring structure of the glycosaminoglycans. Then Benedicts test, because all the test is important, but out of that I will take out Benedicts test, that is very important. And remember, the Benedicts test is positive only for reducing substances and the reducing carbohydrates. Then dietary fiber, very important applied aspect. What is the calorie that we are getting? What are the dietary fibers which are the digestible, like the soluble form, the insoluble form, all these things are very important. Then glucose transporters, I do not have to mention about it because it is a very important applied topic. Glucose transporters, what is its remarks, that is where is it present, in which state it is, like under the influence of insulin, which is the glucose transporter that is active, etc. So, insulin dependent, insulin independent, etc. Then malabsorption disorders, especially the carbohydrate malabsorption, very frequently asked for NEET PG exam, and as well as the glucose absorption, like digestion disorders like lactose intolerance, then about the sucrose intolerance, etc. also question can be asked. Then the pathways, I should say the 4 major pathways. Among that, very important is the glycolysis. So, if you are learning glycolysis, rather than learning the each and every steps of glycolysis, you should concentrate more on the energetics, then what is the substrate level phosphorylation, which are the irreversible steps of glycolysis, the major regulatory features, like it is active in the stage of well fed state under the influence of insulin, that is in the high insulin glucagon ratio, those important points, and Rappaport-Lubering cycle, what is its relationship with cancer, etc. is important. Then gluconeogenesis. So, in the gluconeogenesis also, what are the substrates of gluconeogenesis, then what are the key enzymes of gluconeogenesis, the energetics of gluconeogenesis, very important. Then the next thing is the glycogen metabolism. I will say, I won't say that it is very important, but at least you should know what is the coenzyme for glycogen phosphorylase, that is very important. What is the step that is common to glycogen synthesis as well as glycogenolysis. Such key topics, like the most important topics of glycogen metabolism, rather than learning the whole pathway. Then the applied aspect of it is very important, that is the glycogen storage disorder. Then the HMP pathway. HMP pathway, do not learn that pathway, what you should know, what is the importance of the reversible phase, what is the important, that is oxidative and the non-oxidative phases are there, which is the reversible, which is the irreversible, whether ATP is generated from HMP pathway, what is the significance of NADPH, very important, what are the functions of NADPH, then pyruvate dehydrogenase. The 5 coenzymes of pyruvate dehydrogenase and its clinical application. The deficiency of pyruvate dehydrogenase causes lactic acidosis. So, that clinical aspect is also important. Then glycogen storage disorder, and out of the glycogen storage disorder, I will pick and choose the von Gerke's disease, Curie's disease, then Anderson disease, and among the muscle glycogen storage disorder, I will pick the Pompey's disease, that is type 2, and then the most common glycogen storage disorder in the adolescent age, that is the McArdle's disease. Then in the minor pathways, that is the galactosemia, very important, the clinical features of galactosemia you should be knowing. Then hereditary fructose intolerance and the essential fructoseuria. Then glucose tolerance test and the curve that we obtain, because that can be asked as a image based question. Then the specific dynamic action, SDA is specific dynamic action, otherwise the thermic effect of food or diet induced thermogenesis. That is very frequently asked nowadays, so you should be thorough with what is the specific dynamic action. Then the respiratory quotient. What is the respiratory quotient of carbohydrate? Then high carbohydrate diet. What is the RQ of fat? Very, very important. And don't confuse the RQ and the energy that is released. That is calorie released by the protein, the calorie released from 1 gram of carbohydrates, etc. And what is the respiratory quotient? These two are two different entities. Don't get confused. Then high energy compounds. What is high energy compound and what are the high energy compounds? Then inhibitors of electron transport chain. Among all the things in the electron transport chain, first you should learn the inhibitors, very, very, very important, and TCA cycle. So I've told you, like in any other pathways, rather than completely learning the entire enzymes and the entire flow of the pathway, in the TCA cycle, what is important is where is it taking place, what is its function, then what is the concept of TCA cycle, what is the substrate level phosphorylation, what are the energy yielding steps, what is the energetics of TCA cycle, etc. is more important, and more the anaplerotic role of TCA cycle, and the amphibolic, it's an amphibolic pathway that is very, very frequently asked in the case of TCA cycle. Then the energy content, that is one question that I have given in the first question, that is energy content of food substance. That is also important because depending upon what is the amount of carb, what is the amount of protein and the fat, the energy content of the food is varying. So I have discussed some of the questions in the carbohydrate metabolism, some new trend questions, and also I have discussed what are the must learn topics of carbohydrates. So please, if you have any doubts, please don't forget to ask me. You can put your doubts in the comment section. I'll answer the questions. And if you have any queries, you can ask the questions in my Telegram group or in the Facebook. And if you like it, please don't forget to like, share and subscribe. Thank you.